Yeah, thank you all for having me. And I know today is December 16th, which really snuck up on me as well. So this does not have to be overly formal. Please interrupt me at any time. My slides are kind of a combination of some of my teaching slides, some of my research slides. And Chad didn't tell me that it was the fog project that you guys were particularly interested in. So I'm sorry I didn't put any fog in, but I'm super happy to talk about that. And I can attempt to grab some slides pretty quickly from my student's notebook if I need to. But yeah, we work on lots of different micrometal interactions in our lab or microbe-mineral interactions in our lab or degradations of like lots of different types of processes. Sorry, I'm going to make the Zoom window a little smaller so I can see what I'm talking about. Okay, great. So in our lab, we do waste management kind of broadly defined would be one way of describing it. But we don't really do a lot in wastewater treatment plants. But we work on how microbes interact with contaminants or interact with minerals or interact with saponified fats, oils, and grease in sewers. And we do this in a variety of different contexts. So some of our projects in the lab look at how the cyanobacterial microbial so the cyanobacteria and then their heterotrophic microbial communities that cause harmful algal blooms in lakes, how they form biofilms on marine debris such as plastics, and how that changes the growth of that harmful algal bloom, and also if there's any deterioration of the plastic, and also how that regulates toxin production within the cyanobacteria. So that's one kind of an example of that. I also didn't put slides in for that one today, but I'm happy to talk about it or grab slides quickly. We look a lot at how agricultural groundwater contaminants, which can include, and we like to look at exposures or environmental exposures as mixtures, because we know that a single contaminant alone or a single molecule alone is not realistic of what's happening in the environment. We all get exposed to multiple chemicals all the time. We call it exposome more broadly. Some of our specific projects right now have been looking at these mixtures of using sampling reports from around the state of Wisconsin. We take actual mixtures of groundwater contaminants from wells where people are using those as their drinking water source. And we look at how this mixture of agricultural contaminants can be chemicals, it can be metals, it can be emerging contaminants like microplastics, how those mixtures affect gut microbiome activity and health performance. Oh, that can also include pathogens like salmonella. So we've looked at how salmonella forms biofilms on plastics. And so if there's an exposure to salmonella and to a plastic, that disrupts gut microbiome activity much more than just either one of those exposures alone. So it's really kind of emphasizing that when we're thinking about environmental problems and particularly their health aspects, it's this combination of abiotic processes and biotic processes, but also doing it at environmentally relevant concentrations with environmentally relevant microbial communities, rather than just model microorganisms are all really important to help teasing out some of these processes. We also do some work with bioremediation in aquifers. So this actually relies on a process of microorganisms using their metabolisms to change the solubility of metals. We work mostly on uranium, but this would also apply to many other types of metals. And so we look at how they go in and out of different mineral forms and how we can control that microbial community metabolism in the aquifer in order to have the metal speciation that we want for a target process. Or for recovery or for leaching or all kinds of different things. Yeah, we also have some projects. I'll share some of our work on plastic biodegradation today. with some strains that we isolated from a landfill. I thought that one would be a good example because it shows how the microbes are accessing something that is crystallized. Plastic is just crystallized carbon, but some plastics also have nitrogen in them. So and I was thinking that might be relevant for some of the kidney processes, kind of showing how these microbial inorganic carbon and nitrogen cycles kind of combine to degrade or break down weird minerals. And I should have put the slides in. I should have thought that that's what Chad was talking about was the FOG project. But we also collaborate with the city of Madison on helping get rid of fat soils and grease clogs in the sewers. So this is upstream of the wastewater treatment plant. This is in the sewer lines. And certain places get accumulation of, I describe it that it looks like really nasty cookie dough of just fat soils and grease that accumulate. And we've been looking, and as those persist, they become very mineralized and hard. They react with the calcium through a saponification reaction. And so we've been looking to see if we can use some of our knowledge of mineral metabolism to access those. I'm going to skip that slide for now. Okay, so what I thought I would do is give an overview of microbial mineral interactions. And then I can dive into the example of plastic biodegradation. But like I said, please interrupt me, tell me to skip things if it's boring, ask any questions that you like. I could talk for literally hours about this subject. So I get really excited about microbial interactions. And one really fun thing about being an environmental microbiologist right now, and I actually come from a chemistry background. All of my degrees are in chemistry, but I find that microbes very fascinatingly do all of these transformations of elements, and these are processes that drive biogeochemical cycles, but also have a lot to do with our health. And there are also processes that we have the potential to control, both as therapeutics or, as therapeutics for the environment, as it were, with some of these bioremediation things. So this little diagram I grabbed is just an overview of different types of microbe-mineral interactions that can occur. Environmental microorganisms have a huge range of metabolisms, and we like to capitalize on those metabolisms. So this is just a subset, but some of the big categories of micromineral interactions are things like bioaccumulation, which would be where a mineral accumulates inside of a cell. So microbe is uptaking that mineral or a portion of that mineral usually for nutrient reasons. There can be absorption of, so there's different types of absorption. So a microbial cell has all of these layers on the outside with different, EPS or different, which stands for extracellular polymeric substances, or all these different layers, pili, flagella, all these different kinds of things. So there are many absorption places on a cell. And if you really want to get fancy, there's a difference between sorption, adsorption, and absorption, but we won't go to those levels of details today. But suffice to say that you can have minerals interacting with the surface of microbial cells in many ways. Sometimes they just stick to the surface. We call that biosorption. Sometimes you have chemisorption, or you can have these biomineralization processes where as a metal or or any molecule comes closer to the cell, a chemical reaction takes place that's mediated by a product that the microbe is making, and you get these minerals that formed and they stick next to the cell. You can also have things like metal chelation. So one of the ways that microbes obtain needed metals or other nutrients for their environment is by producing what we call chelating molecules, which are small molecules that go out and act like a stapler, and they grab whatever nutrients that they're looking for. So you get these metal chelating reactions. That's another way. There's other things like biotransformation, where a microbe is directly acting on the mineral and usually changing the redox state from oxidized to reduced or reduced to oxidized. It can go either way. We call that biotransformation. And then there's other examples of things like bioleaching, which if you picture microorganisms, most microorganisms on the planet live in anaerobic environments. We tend to think in a very oxygen-centric viewpoint because we need oxygen to breathe and we do oxygenic respiration. But most microbes don't. They live in anaerobic environments that are very nutrient limited. And so they have all of these different techniques in order to access those very difficult to get at nutrients. And so one of those is by weathering or dissolving minerals to release either the metals or the carbon or the nitrogen or the sulfur or phosphorus or whatever it is that they need out of it. And we call that bioleaching. One of the most common ways of doing bioleaching is that a microbe will secrete an organic acid, usually a fermentation end product, to actually use a low pH to dissolve the rock or the mineral to release the metal or whatever it is that they're looking for. Like I said, if I'm being too high level, just stop me at any time. So these are some like specific examples that I was going to show. So some of them we call like acidolysis, where they secrete a bunch of acid to release the metal. You can have redox reactions. These get used by biomining companies, which there's a huge uptick in biomining companies right now because of critical metals recovery and other types of priorities. And then we also see a lot of this in agricultural applications for producing better fertilizers for plants, in situ in a soil environment. And to kind of zoom in on some of those soil processes, which could be quite similar to what might be happening, in a kidney or in a urinary tract situation, is where you have microbes producing inorganic acids or organic acids. Those can weather or break down the minerals, which release all kinds of things, calcium phosphate, potassium, iron, etc., sulfates, and then you have the microbes uptaking and metabolizing those and then helping to feed the plants. Those can also be driven by siderophores, are the most common type of those metal chelating molecules, and then you can also have some of these redox reactions. And so I think there's a lot of possibility for like kidney situations because microbes have been naturally doing these types of mineral weathering and breaking down processes as a natural part of their metabolism for a very long time. Just to briefly distinguish, so of course, not all mineralization is biologically driven, but a ton of it is. So you can have just regular mineralization, of course, abiotic processes. And then you can have, so microbes can both make minerals and break down minerals. I'm showing some examples right now of microbes making minerals, and they can do this usually on their outer membranes or in a lot of different ways. And we usually break those down into two bigger categories that are roughly called biologically controlled mineralization, where there's a specific mechanism where a microbe controls the biosynthesis of a mineral for a very specific purpose. I'll show you a couple of examples later. One of my favorite, I do have it on a slide, so I'll wait. But one of my favorites is called magnetotectic bacteria. And they synthesize magnetic nanoparticles inside of their cell. And then that's what they use to swim. It's crazy. And so those are really fun. And so, yeah, there's these biologically controlled mineralization ones. And then potentially what might be more relevant in a urinary tract situation is this biologically induced mineralization, where microbes are producing organic acids or inorganic acids. They also, have all these sticky things outside of their cell, and that's creating an environment where you can have these abiotic mineralization reactions happen. These can also be enzymatically driven. Many organisms have phosphatase enzymes or things like that on the outside of their cell to create phosphates or sulfates such that they complex with calcium and form these mineral aggregates. So lots of different reactions. Maybe I'm showing you all too many, but I was trying to, these are actually some of my absolute favorite examples of biomineralization from microbes. And so these are all of these, except with the exception of E, those are our magnetotectic bacteria, but all of these are different microalgae that form calcium carbonate or calcium or silicate structures as like the physical armor around their cells. So all of these are microscopic, they're very tiny. They still make them, and we can see these persisting for a very long time in the fossil record because they're these inorganic structures. And so microbes are capable of not only randomly making minerals, but they're capable of making highly structured and highly ordered and highly organized mineral structures that they use for a variety of purposes. So for mobility, for protection, for nutrients, it's just this whole wide world of why microbes make minerals or why microbes interact with minerals. And so, yeah, these are some of my absolute favorites. I just think they're just such beautiful microscopy images. And the microbes completely control that structure with different proteins and select biosynthesis. So you can get very complex architecture of minerals as well. This is an example of a less common one, but oh, I did put in a calcium thing. I knew I did. All right. So this is an example of how a microbe can facilitate a calcium phosphate or calcium carbonate mineral forming. You would get a very similar process with calcium sulfate or calcium phosphate. So you have a microbe that produces carbonate or phosphate or sulfate, and those get exported externally from the cell, and they can react with calcium in the environment to form precipitates that tend to aggregate or stay near the microbial cell because of that. Oftentimes, these are driven by inorganic nutrient metabolisms, like the urea cycle, for instance, which I also put in here because this was an example that I grabbed because I assumed that urea would be quite relevant in a urinary tract situation. So many microorganisms are capable of using urea as a nitrogen or an energy source. And so that can drive some of these precipitation reactions. The carbonate kind of comes off as like the off-gassing of the urea respiration. Okay, yeah, how long am I talking? All right, so these are some other favorite examples of mine. These are the magnetotactic bacteria. So they tightly control the synthesis of these magnetic iron nanoparticles inside of their cells. They're arranged just like the needle of a compass, and it orients the microbe to the magnetic field of the earth. I know this sounds crazy, but it works. You can control them with a magnet, and then that's how they have their motility. And the biosynthesis of these clusters is very well worked out. Another example of external sometimes, but important microbe mediated mineral reactions is manganese oxidizing bacteria. So yeah, microbes can create all these manganese minerals and so yeah, they do the same thing, but this is for energy generation. So they're oxidizing the manganese to get the electrons out to drive their energetic processes. So they form these really highly ordered crystals as well. Okay, there we go. And then maybe of interest to this group as well is These are some other examples of how microbes can break down minerals. One way that is called is just bioleaching. Other words would be biodegradation or things like that. I apologize, the slide is really busy. I couldn't decide whose image I wanted to steal, so I threw all of them on here. And I'm happy to provide all of the photo credit references. Sorry, I realize those are in my notes, not on the slide where you can see them, but I'm happy to share those. So we have a lot of different ways that organisms can erode minerals. A lot of them are sometimes what we call like accidental, where it's the end product of a microbial metabolism that happens to be corrosive, like hydrogen sulfide, and that can just degrade or weather or corrode a mineral. And so you have that kind of breaking down. Others are more intentional where, again, they're secreting organic acids or doing these other ones to get at specific ones or to get at specific nutrients. So a lot of companies have actually started to kind of capitalize on this to recover metals from mine tailings or other opportunities like that. And so, I also put this on the slide to say that these processes don't have to have direct contact, so they can be mediated by microbial metabolites or end products that are free in solution that cause a reaction with the mineral. So it doesn't always have to be a direct contact reaction. Okay. So I grabbed these really quickly. I will be excited to dig into these and learn more, but I've seen over the last several years that there have been people starting to study if microbes have a role in the production of kidney stones, and then also maybe in the dissolution of kidney stones. And so I think there's some really exciting research kind of supporting that, very similar mechanisms to what we see occurring in the aquifers that I look at, or in the landfill like I look at, could be very similar types of processes that might be happening in the kidney. Okay, so I will probably go fairly quickly through these or maybe skip over some of these slides. The next slides are kind of from my class. I was going to dive a lot into the microbial metabolism of them, but I think I said a lot of the points I wanted to. So I'll go through these really quickly just to skip ahead to the example of plastic biodegradation. So I think I was just trying to make the point that these micromineral interactions are driven by microbial need for energy and or nutrients. Remembering that most microbes, including those in the urinary tract, are living in a very low to no oxygen environment. And so they need to use these diverse metabolisms to access these essentials. So or places where you don't have access to organic forms of carbon, nitrogen, oxygen, phosphorus, et cetera, where you need to get those essential nutrients out of inorganic materials. And microbes have been doing that since the dawn of time. So, or the dawn of microbes, rather. Sorry, not a geology talk. Okay, so I always like to put this in here just to remind us that microorganisms or biological organisms in general are not just organic things from this part of the periodic table, that we have a lot of inorganic things in us. Yes, even cobalt, yes, even vanadium. You have enzymes inside of you that use vanadium. It's really fun. So, we have, and you have tungsten containing enzymes also, very essential for you. So we're not just talking about carbon, nitrogen, oxygen, phosphorus. We need all of these types of minerals. Those minerals and trace metals and things, they all get accumulated into all of our cellular structures. And microorganisms in particular are really good at using a huge variety of carbon sources. So they can use inorganic carbon in a variety of forms. They can do fermentation. And there are even types of fermentation that only use amino acids. So you can just straight up ferment an amino acid, which I think would also be relevant in a urinary tract environment. So you get all these different types of carbon metabolisms. So depending on what carbon is available, you can enter and exit central carbon metabolism at any point, and there are multiple entry and exit pathways. So we always draw things from a very glucose-centric point of view, but biology doesn't see that way. You can enter here at isocitrate, you can enter at septohellulose, oh my gosh, cetoheptulose. I flipped my consonants on that word. You can enter wherever you want. And so yeah, when we're thinking about a place like the urinary tract where you have, I'm assuming, very limited resources, these types of unusual microbial metabolisms, both in terms of carbon source use or sulfur source or nitrogen source use, as well as these unique ways of getting energy from, I already talked about, there can even be amino acid fermentation. There are many, many different types of anaerobic respiration that all help drive these processes. A lot of these processes evolved because of stratification in the subsurface. So you have different, as you have an oxygen gradient, you also have a redox gradient, which makes different types of metal transitions possible. And this promotes different microbial metabolisms that exploit the energetic differences between the two different forms of a metal. And they use that as their terminal electron acceptor in their respiratory chain instead of oxygen. So they're manipulating metals to do that, which is why they need to access the minerals to do so. These are other examples of stratification of the subsurface. I think a particularly relevant form of this type of metal metabolism when we're talking about minerals or perhaps something in a urinary tract would be this extracellular electron transfer. So these are types of mechanisms that microorganisms can use to access electron acceptors and donors that are extracellular to their cell and also much bigger than their cell. So they have both direct contact ways and indirect contact ways of interacting with that mineral surface in order to get the energy or the nutrients out of it. So we call that extracellular electron transfer when a microbe is accessing those electrons from external to the cell. They're really good at that. Nitrogen cycle will be super important, I think, in a urinary tract environment, as it is in any environment. And so we would just want to look at how nitrogen is cycling within your specific environment, not all over the place. But I just left that in there to say that there are many types of nitrogen metabolism. So microorganisms exist that can modify pretty much any form of nitrogen, whether inorganic or organic, and they can oxidize or reduce them. So you can go any which way between these different forms of nitrogen. There are microorganisms that exist that can carry out all of these, which includes urea. The urea cycle is a little bit different, but it enters slightly differently into this pathway. So yeah, so for microorganisms, not a problem. Similar, same thing with the sulfur cycle. You have microorganisms that can modify sulfate all the way up to sulfide and everything in between going back and forth. These processes cause a lot of problems on the planet, like biocorrosion and oil souring, which would be kind of analogous to some of the challenges in different human health environments. And there's already a lot of evidence of things like sulfate-reducing bacteria that cause hundreds of billions of dollars worth of corrosive damage a year. These similar processes happen in human gut microbiota that have a lot to do with sulfide signaling and inflammation related to different diseases in the gut. I was looking at uranium precipitation, and we still look at uranium precipitation. I'm going to skip ahead to plastics because that's, I think, a little more relevant than metal precipitation today. But I'm always happy to talk about uranium. And a lot of that is mediated through what we call microbial influenced corrosion, which has a couple of different pathways that I've kind of already loosely introduced. But you have some type of inorganic energy metabolism, releases corrosive things like hydrogen sulfide that can cause pitting on a surface or cause the formation of iron sulfides. And so you get pits in one area and piles of iron sulfide minerals in other areas. So yeah, all kinds of all kinds of stuff going ahead there. Okay, so I'll show you an example that we've been working on in my lab lately on plastic biodegradation. Also happy to talk about fog biodegradation or anything that would be useful. So we got into plastics because we're interested in this idea of circularity for materials where we would ideally have no waste. So you can convert one product into something else and have that kind of infinitely circle instead of ending up in a landfill. So plastics notoriously not a circular material. They've been causing a lot of pollution, and they're also a health risk because of the pathogens and other toxins that can bind to their surface and then be transported. Plastics are very challenging to remediate in the environment because they dominantly end up in aquatic systems, but they don't have a point source and they're very dispersed. They have many different densities, so they don't stay in the same place in the water column. And so it's very tricky to have bioremediation for these. So we'd like to ideally replace them. And in my lab, we did start thinking about places we could intervene, like wastewater treatment plants. But today I'm showing you more of our biodegradation side. So of course, not all degradation in the environment is biologically driven. There's a lot of abiotic processes that can help stimulate or speed up biological degradation processes. For plastics in particular, sunlight is a big one that helps start to make the plastic more brittle and fragment into other pieces. The other big thing that sunlight plus oxygen does, or interactions with water, is that it causes oxidation of the surface, meaning that you now have different functional groups on the polymer, which makes it easier for microbes and enzymes to start to attack. So when we think about microbes degrading plastic, and you could almost replace this with any type of like mineral or fat deposit or things like that, is that there's generally these types of steps. And I'll point out that the first few steps, the hardest ones, have to occur extracellularly, because usually what's being degraded is bigger than the microbe itself. So the microorganism has to secrete the enzymes or the small molecules or whatever out into the environment and hope that it reaches the right target in order to start those breakdown processes. And so it actually takes quite a bit of effort and steps to have that product be small enough such that it can be transported or uptaken inside of a bacterial cell or a microbial cell. So crystalline breakdown for plastics, that can be abiotic in nature, but it can also be biotic. So we started kind of thinking about, for all of these different types of steps for plastics, can we go out in the world and find microbes that did it? This was one of our pandemic projects where we were just looking at the chemistry of how these processes would have to happen. And we made some predictions about the enzyme function that would be required to target different types of plastics. We then got the opportunity to sample the local landfill here in Dane County. for microorganisms throughout the site. We've been doing a lot of other things with that, but one thing we did do was look for potential plastic degrading microbes. We found that throughout the landfill there were many potential plastic degrading bacteria, but there were way more potential plastic degrading fungi. I don't know if fungi are relevant to the URI microbiome or the urinary tract microbiome, but that's a question I'll have for you. I would love to know if fungi are, because that brings in a whole different aspect of biodegradation possibilities. Fungi are really, really good at breaking down minerals. Bacteria do it super well also, but fungi have completely different mechanisms that can be helpful. This is a different way of visualizing that plastic biodegradation process, where you have microbes either colonizing or living very close to the plastic polymer surface. They're secreting those enzymes or organic acids or bases that are starting to reduce the crystallinity, starting to put functional groups onto the polymers, starting to do some depolymerization to the point where those oligomers or small chains of monomers and that plastic polymer are small enough that they can be brought inside of the cell and then further degraded and used as a carbon source as part of like regular central carbon metabolism. It really bothers me that the general term used here is mineralization. So if a microbe gives off CO2, they call that mineralization. I really hate that, but anyway, that's a sidebar. We know that microbes can degrade plastics, but the plastic structure is really one of the limiting factors. It's this crystalliness of the plastic. Microbes are perfectly capable of metabolizing the polymer, but once it's into that crystalline form, the enzymes can't access the right orientation to do the depolymerization reaction. So the microbes have to use all of these bioleaching or these corrosive or mineralization processes in order to break the crystalline structure up enough that they can release those polymers. And we see that polymer structure is directly related to predicted biodegradability. So some plastics are, because of their structure, are much easier to biodegrade than others. So things like polypropylene, polyethylene, which are carbon-carbon backbones, very tight crystals, those are very hard to biodegrade versus things that are more flex or less crystalline and have some heteroatoms like nitrogen or oxygen in their polymer backbone chain that is easier for microbes to break down. Sorry, I'm just seeing the time, so I'll skip a little ahead here. There's not a lot known for some of these biodegradation pathways. If we zoom in here, we would see that says pyrolysis, which is definitely not a biodegradation step. Pyrolysis means heating at high temperature and high pressure. I think we still have a lot to learn about them. That being said, there are certain classes of plastics where we have found enzymes that do act on the intact plastic. The water bottle plastic, PET, polyethylene terephthalate, we do now know the sequence of enzymes that can break this down, but this is a polyester, so much easier to break down. Same thing, like there's some examples for like polyurethane where the enzyme is known that can do that reaction. So we went into our local landfill and we grabbed plastics and soils, and we enriched these microbes for growth on plastic as a sole carbon source. We used a combination of polyethylene, polystyrene, and polyamide. We do see that over time, or from the different places in the landfill, when we grab plastics from different ages of the landfill, we found that those microbial communities were very similar to each other, even though the microbial community found in the soil surrounding that piece of plastic looked completely different. So that's suggesting to us that that if a microbial community is growing on a piece of plastic, that it's selecting for a specific membership or a community of microorganisms that collectively contain all of these enzymes that would be needed to do plastic biodegradation. As you saw, plastic biodegradation requires a lot of steps, so we kind of predict that it will be a microbial community that is best at actually doing plastic biodegradation. So we wanted to see more specifically for some of our communities who was there and how they were doing it. So we set up a series of more specific enrichment cultures, and we separated the plastics. So we were looking at each plastic individually. We saw that we could get a microbial community to routinely grow on nylon 6 or polyamide. And we're like, great, this is exciting. One of the graduate students in the lab, David, did have the good idea, though, to do a hot water extraction of the polyamide before we put it in, because plastics are not just the polymer. When plastics are made, they have all these small molecules that we call plasticizers that get incorporated into them. And so David then grew the microbes on just the extract alone, these additives and small molecules, and found that they grew very well, and they grew not at all on the extracted polymer. So this was kind of a sad day in the lab a little bit. So it told us that our microbial community was preferentially eating the small molecules and not eating the polymers, which makes sense. A microbe is only going to access those difficult to reach carbon or nitrogen sources in the plastic when it's really desperate. And if anything else is around, it's not going to do it. And even in those cases, it can be so hard to access that it won't. But I wanted to show this because this is an example of unusual nitrogen metabolism. And so David did follow up to see what molecules were in those extracts, because we were just using a commercial plastic, so we had no idea what those extracts were. So we identified them with GCMS and NMR and found that it was this caprolactam molecule and then its cyclic dimer. And we did a bunch of growth experiments with our microbial community, and we showed that if we grew it on minimal media and provided no nitrogen and no carbon except for the extract molecules, that the microbes grew very happy. So that was telling us that they were capable of extracting both carbon and nitrogen exclusively from caprolactam, which is what we often see with these degradative metabolisms. So we weren't the first person to see some of these metabolisms, but we have been thinking about how we could use them, and we've been characterizing that pathway. We isolated one of the organisms that seems to be contributing to that, and that was recently published, which is fun. We also have a collaboration where for some of those waste plastics like PET, where we've been able to engineer this into thermophilic microorganisms to degrade PET and then funnel that carbon actually into producing a biodegradable plastic polymer, which is really fun. And yeah, I think I'll stop there and try to take any questions. That was a little bit of a tour de force through a lot of micro-mineral stuff. And so hopefully that made sense. And yeah, I'm happy to talk about any of those processes.